Well, so the last class, not exam, but the class before that, we learned about our non-specific defenses. Remember, we have two lines of non-specific defenses. Our first and second line. First line defenses are things like skin, mucous membrane, stomach acid, things like that. Our second line defenses involve cells like phagocytes that eat things, also inflammation and fever. So if a pathogen gets through or gets past the first and second lines of defense, we do have a last line, so a third line defense, and that is our adaptive acquired immune response. And this response is very specific to that particular pathogen. And so today we're going to start learning about how we create that specific immune response. But it is a war, right? So it's a war for survival. If the pathogen gets through the first and second lines, you have this last line, and it's all or nothing. And so your immune system is going to use everything it has to attack and destroy that pathogen. In the process, there's sometimes incidental host damage. As I've already mentioned, we'll talk more about that today as well as next class. So your adaptive acquired immune response involves antibodies, lymphocytes, macrophages, all kinds of things coming together, right, to attack that foreign thing, right, that pathogen, that antigen, whatever it is. So antigens are what the immune system recognizes as foreign, right? They do not belong. And so the immune system will attack that antigen. So anything that stimulates the immune system is an antigen. And the typical response is the production of antibodies. And we're going to talk a lot about antibodies today, right? They're special immune proteins which combined to an antigen in order to neutralize it. And once again, we'll talk about complement. Because remember, the classical pathway to activate complement is by using antibodies. So your adaptive acquired immune response is something that you acquire during your lifetime. So every time you're exposed to a new pathogen, you create an immune response against it. So you acquire, right, this immunity as you live, as you encounter new pathogens. The benefit to acquiring this immunity is that it provides you long-term protection, usually for the rest of your life. But as we mentioned before, pathogens mutate and change. And so your immune system has to adapt to that by creating an all-new response against that mutated pathogen. So even though you have this long-term protection, you may never encounter that exact same pathogen again because it's mutated and changed just enough so it looks brand new to your immune system. And we are going to see that the adaptive acquired immune response involves a complex interaction of antibodies, T cells, B cells, macrophages, chemical mediators, complement proteins. So as I said, your immune system is going to use everything it has, right, to fight off that pathogen because it's a war for survival. Now, we're going to cover the key components of the immune system. Not every little thing, because you can actually take entire semester-long courses on the immune system. So we're just covering it basically in a little over one lecture. So there's a lot more to it, but we're covering all the major components and the major interactions. So about half of your immune response is called humoral immunity, and that involves antibody production. And it's the B cells or B lymphocytes that create the antibodies. And as we'll see, the B cells actually have antibodies attached to them, and those antibodies act like a receptor or an antenna, right, to interact with antigen. But it's the B cells that make the antibodies. The other half of your immune response is T cell mediated. And so we will talk about a few different kinds of T cells, right, to see their involvement in the immune response. But it's a complex interactions of T cells and B cells and macrophages all working together, right, to fight that pathogen. So we have some terminology. Serology is the study of antibodies and antigens. An antiserum, as we mentioned before, is a preparation of antibodies. If you take blood and you centrifuge it, you separate out the red cells and white cells, and the liquid portion is the serum. And the serum contains proteins. And so we call serum proteins globulins. And the antibodies are immunoglobulins. And that portion of the serum containing antibodies is called the gamma globulin. And the antibodies are going to bind to antigens, right? Those foreign substances that trigger an immune response. So what kinds of things are antigens? Well, proteins and polysaccharides are the most antigenic substances, right? So proteins and polysaccharides are very antigenic, but even lipids and nucleic acids can be antigens. Now, when it comes to antigens, size does matter. And don't worry about this exact number because it depends on what source you're looking at. But the main idea, if an antigen or if a substance is too small, it's not an antigen because the immune system cannot see it. So the antigen must be sufficiently large so the immune system can see it and react to it. Now, some antigens, like some large proteins, they're so big there are different parts of that protein that can be seen by the immune system. So we call those different parts epitopes or antigenic determinants. So it's possible if you have a very large protein, your immune system can actually make different antibodies to attack that large protein because each antibody is attacking one specific region of that protein, right, the epitope. Now there are substances called haptens, and these substances are not antigenic by themselves. I like to call them antigen wannabes because they want to be an antigen, but by themselves they're too small and insignificant. But what can happen is that if the hapten gets stuck to one of your tissue proteins, that combination of hapten and protein looks foreign to the immune system, and that can trigger the immune system to attack. But these are usually inappropriate responses where you do not want the immune system to attack because the immune system ends up doing damage to the host. And we'll talk about those kinds of reactions next class. But things like penicillin allergies. Some people are allergic to penicillin, most people are not. But somewhere along the line when someone was taking penicillin, it got stuck to one of the proteins in their serum, and the immune system saw that protein penicillin combination and did not like it. And so they made antibodies against penicillin. So in the future, if they were to take penicillin, they could have a severe allergic reaction which could kill them. And we'll talk about those next class. Poison ivy. If you've ever gone out playing in the woods and then a couple of days later you break out in a rash and blisters, it was caused by the urushiol oil in the poison ivy plants. And the urushiol oil is a hapten. But in some people, it combined with a protein in their skin, and their immune system made a reaction against that. And so that reaction, causes the destruction of epithelial cells, of skin cells, and that causes blisters. So again, it's an inappropriate reaction. You do not want this to happen, but it does happen. So haptens, antigen wannabes, small and insignificant until they combine with one of your body proteins. And then that looks foreign to the immune system. Usually causes inappropriate reactions, right? Things that you do not want to happen. But again, we'll talk about those next class. So you can think of a bacterial cell, for example, as an antigen, but in fact, its cell surface is covered with proteins and polysaccharides. So there are many antigens associated with that pathogen. If you have a large antigen, there are different parts of that antigen that can be recognized by the immune system. And so the immune system can make antibodies against those different parts. In this cartoon example, antibody A, it specifically binds to the triangular shaped antigen. Antibody B binds to the round one. So these are two different antibodies, and they bind to two different epitopes or two different antigenic determinants. And so this is partly why you get that long-term protection, because your immune system is not simply making one kind of antibody to that pathogen, it makes multiple kinds of antibodies against that pathogen. And we call this kind of response polyclonal. So a polyclonal response. So you're making more than one kind of antibody. Now, has anyone heard of monoclonal antibodies? Anybody ever heard of monoclonal antibodies? Yes, Rachel. So do you know anything about them, or you just heard them?
No, I've heard that word.
Yeah, so often we hear this in relation to cancer treatment, because some of the newest therapies involve using monoclonal antibodies. So when it comes to cancer cells, all of us occasionally have cancer cells that pop up in our bodies, and the immune system quickly eliminates them, right? So it's very common for abnormal cells to occur, and the immune system will destroy them. The problem happens when the immune system cannot see the cancer cell as a cancer cell. It cannot see that cancer cell as something bad, right, that needs to be destroyed. And so the immune system just ignores it because the immune system can't see, hey, oh, this is a bad cell type, I need to destroy it. And so the idea behind the monoclonal antibodies are a signal to the immune system to attack and destroy. So what they do is they find an antigen unique to that cancer cell. So that antigen is only found on that cancer cell, nowhere else in your body. And they make an antibody specific to that antigen. So when they inject you with the antibodies, they only bind to the cancer cells. And that alerts the immune system to come attack and destroy those cancer cells. So that's the idea behind the monoclonal antibodies, is you're targeting the cancer cells so you can alert the immune system to say, hey guys, come over here. Here's a cancer cell. You need to attack and destroy it. But the antigen must be unique to the cancer cell and not found anywhere else in your body. Otherwise, the antibody would stick there, and then your immune system would attack those other places. So, but these are new, very impressive therapies that are being used to treat certain cancers. And so they show a lot of promise because the idea is you're simply alerting the immune system to do its job, right? To attack and destroy cancer cells, which your immune system normally does very efficiently. But every once in a while, the immune system just cannot see that cancer cell as a bad cancer cell. But we do tend to make multiple kinds of antibodies against a pathogen, so that guarantees that you're going to have at least one of those available, during your lifetime that can attack that pathogen again, and that helps to provide that long-term protection. So antibodies are Y-shaped molecules. They're actually shaped kind of like a Y. Here's a cartoon illustration to show you the overall structure. There are two long, heavy chains of protein and two short, light chains of protein. The regions labeled C are constant. So what that means is they are common for all the different kinds of antibodies. What is different are the variable regions on the tips. These are the regions that are different for every different antibody because they must bind to the antigen. And remember, the binding between molecules involves chemical bonding, right? Covalent bonds, ionic, hydrogen bonds, van der Waals forces. So in order to recognize any given antigen, you need one type of antibody that can match that type of antigen. So the variable region is different from antibody to antibody. But the constant region is a common backbone structure for all the antibodies. A little bit more realistic model is a computer-generated image here. And again, it's a Y-shaped molecule with the antigen binding sites on it. There are 5 classes of antibodies, and you need to be familiar with all 5 classes. And there's a fairly easy way to remember them. What mnemonic device can you use to remember the 5 classes of antibodies? What's a way to remember IgG, IgM, IgA, IgD, IgE? How are you going to remember that for the exam? GMADE, right? So GMADE is the way that all students used to remember it. Right, so GMADE. So let's talk about each class. So IgG antibodies are monomers. This means there's just one Y-shaped molecule. And when you look in the blood serum, the majority of antibodies present are IgG antibodies. They have the ability to fix complement. That means to activate the complement cascade, right? The classical pathway where those serum proteins associate to poke holes through cell membranes. And very importantly, they cross the placenta. So this is important because that developing fetus has no immunity, right? It's still developing its immune system, so it has no immunity. So mom's immunity can actually cross the placenta to protect that developing fetus. So the IgG antibodies are able to cross the placenta. So mom's immunity can help protect that developing fetus. Now, the first type of antibodies actually created, right, the first time you encounter a brand new antigen, are IgM. And IgM antibodies are big, right? They're actually 5 antibodies all connected together. So it's a large antibody molecule. And one of the advantages of producing the IgMs first is they have a lot of antigen binding sites, right? You have 10 potential antigen binding sites. So an IgM molecule can bind up a lot of antigens all at once. IgMs are able to activate the complement cascade, but they are too large to cross the placenta and protect that fetus, so they don't do that. But the first type of antibodies to be produced upon the first encounter with a brand new antigen. Later, your body will switch to making just IgG antibodies, which it can make a lot faster, right? Because they're just simpler molecules. IgA antibodies are dimers, so there's 2 of them stuck together. And we find these on mucous membranes. So they help to protect the mucus or the mucosal epithelial cells, right? So they exist. And remember, we talked about one possible virulence factor produced by some bacterial pathogens, an IgA protease. So some bacterial pathogens produce an enzyme that can destroy the IgA antibodies. And that will help that pathogen to survive and infect, right, the mucosa. IgD antibodies are found in the blood, lymph, and on B cells. So remember, I said the B cells have these antibodies stuck to them, which act like an antenna or a receptor to bind to an antigen. And we'll see that later on. But the last ones, the IgEs, are the ones that I hate. And I hate them because not because of their normal function. Their normal function is to help protect against large parasites like worms. What I hate about them is that they're involved in allergy responses, so in allergies. And one of the things that they do that we'll learn about next class is they cause the release of histamines. And histamines are one of our chemical mediators. So histamines are important, but only when they are secreted at the right time, in the right place, in the right amount. And the problem with allergies and IgEs is that, things like tree pollen cause the massive release of histamine. And that's an inappropriate response that causes the symptoms of allergies. So people who have a lot of allergies, especially in the springtime, we have a lot of IgE antibodies, and we don't need them. But we'll talk more about that next class. So we have 5 types of antibodies, and typically antibodies are made in response to an antigen. But I do want to mention we do have some antibodies that are called natural antibodies, and what that means is they're actually always produced, regardless of if an antigen is present to cause them to be produced. And these are antibodies that sort of evolved with antigens that are always around, that we always had to deal with. And so evolution decided that an organism like us should just always make these kinds of antibodies no matter what. All the rest of the antibodies are normally made in response to a particular antigen, but there are certain kinds of antibodies called natural antibodies that are always produced, right? So they're constitutively produced. I just want to mention that because normally we talk about antibodies being made in response to a specific antigen, but in fact there are some that we always produce all the time because it was necessary, as a result of our evolution, because we encountered these all the time or frequently in the environment, so we always make them. But just to mention that we do have these things, natural antibodies that are always being produced no matter what. But most of the time the antibodies are the result of a specific response to an antigen. And so we make antibodies specifically in response to that antigen when it is present. So since we do make a lot of different kinds of antibodies, they're obviously very important. So there are 5 different mechanisms of antibody action. One of them is neutralization. So if a pathogen is producing a toxin, antibodies can bind to the toxin and neutralize it, so it's no longer toxic. When we talked about viruses, the first thing that any virus must do is attach to host cells. But if the virus is covered with antibodies, it cannot attach. And so the virus has been neutralized. We've talked about antitoxins, or preparations of antibodies against toxins. So things like snake venom, spider venom. We have antivenoms, or technically antivenins or antivenines. So an antivenom is a preparation of antibodies. So if you get bitten by something poisonous and you get to the hospital in time, they can inject you with the antivenom. And those antibodies will bind up the venom, neutralize it so you survive. So let's say you're out hiking one summer in the woods and you get bitten by a rattlesnake, but you're able to get to the hospital in time so they inject you with the antivenom and you survive. You need to be really careful in the future to not ever be bitten by a rattlesnake again, Because if you're bitten a second time, you may not be able to receive the antivenom. And the reason why is those preparations of antibodies are not human antibodies. They're made in other animals like horses. So they're foreign antibodies. And what does your immune system do to anything foreign? It attacks. But it does take time, as we will learn. So those foreign antibodies are able to do their job, neutralize the toxin, and then your body will get rid of them, right? Will destroy them. But your immune system may have become sensitized to those foreign antibodies. So if you receive them again in the future, you could have a massive allergic reaction that could kill you. So that's the problem with an antivenom, is that they're not human antibodies. They're animal antibodies. They're foreign antibodies. And so you may react very badly to them if you receive the antivenom more than once. Oh no, rabies is a vaccine that they inject you with to help stimulate the immune system to fight the virus before it has a chance to really multiply and cause any serious damage. So the rabies vaccine is not a pleasant vaccine to receive, although it's improved, right, since its original creation back in the late 1800s or early 1900s. So it's better now than it was, but they actually use weakened rabies virus, right, to inoculate you, right, to help to protect you in case you were bitten. Agglutination is another mechanism. And so to agglutinate means to clump together, right, to stick together. And so when you have bacteria, one of the things that they want to do is spread. But if they're all stuck together by antibodies, they're all clumped together, so they cannot spread. So this helps to, stop them from spreading. Also, it makes it easier for a phagocyte to come along and eat a clump of bacteria rather than individual cells one at a time. Precipitation involves soluble antigens. So remember, soluble means that it is dissolved. So a phagocyte cannot eat something that is dissolved. But if an antibody binds to that soluble protein, it becomes insoluble. It precipitates. It becomes solid. That's something that a phagocyte can now eat. Opsonization, I always use the analogy of frosting on a cake, right? Because most people love frosting on a cake. It makes the cake taste so much better. And there are some weird people out there that don't like frosting, but most of us do. So the idea here is, if something is covered with antibodies, that makes it tastier, more appealing for a phagocyte to come along and eat it, right? So covering something with antibodies encourages phagocytosis. So like the frosting on a cake, you want to eat the cake more because of the frosting that's on it. And once again, we have complement. So complement fixation or complement activation involves those serum proteins. If antibodies bind to a pathogen like a bacterial cell, it will activate the complement cascade. Those serum proteins will cut and cleave and associate in order to form a protein tube that pokes holes through cell membranes. But the classical pathway, the most common pathway, is mediated by antibodies. Now, in general, complement tends to be more effective against gram-negative bacteria and less effective against gram-positives. Any idea why that would be? Why gram-negatives are killed more easily by complement proteins than gram-positive cells are? Right. When it comes to the cell wall, remember that the gram-positive bacteria have a very thick layer of peptidoglycan, so it's harder for the complement proteins to assemble and poke through those cells. But the gram-negatives, their cell walls are basically all membrane, right? There's very little peptidoglycan, so it's easier for the complement proteins to poke through, right, the lipid membranes of gram-negative cells. So just to summarize all the different mechanisms, including one that I'll mention later, the antibody-dependent cell-mediated cytotoxicity. We will talk about that later on, probably not till next class. But antibodies are clearly extremely important. They do most of the, of the job when it comes to alerting the immune system or inhibiting or stopping pathogens. So antibodies are clearly extremely important. So whatever the mechanism, the binding of antibody to antigen facilitates the destruction of that antigen. So antibodies are extremely important, right, for the normal functioning of the immune system. And that's why we make a lot of them. So antibodies are produced by the B cells, by the B lymphocytes. Remember I said that the B cells have the IgD antibodies stuck to their cell membranes, and they act as a receptor or an antenna. And so they can interact with the antigen, which will cause that B cell to start making a lot of antibodies. As we'll see later, helper T cells really enhance this whole process and are very important at helping to stimulate B cells to make antibodies. But what is interesting, if I were to ask you how many different kinds of antigens are there on the Earth? Any idea how many different kinds of antigens there are? A lot, right? So let's just say for the sake of argument that there are 1 million antigens. And in fact, there are way more, but let's just say there's 1 million antigens. So that means you have to be able to make 1 million different antibodies, right? One for each kind of antigen. So when it comes to antibody genes, that would suggest you need at least 1 million antibody genes, right? But who remembers from the quorum sensing video approximately how many genes do humans have? Everybody's working on the quorum sensing, which is due today, right? By the end of the day. So how many genes approximately do humans have? I know a few of you have already turned in the quorum sensing, right? Does anybody remember how many genes humans have approximately? Oh, no, Rachel is correct. Around 30,000 genes. So around 30,000 genes, human genes. So there's no way we can have 1 million different antibody genes. And in fact, we only have a handful of antibody genes, so it's a small number. So how can we generate millions of different kinds of antibodies? Well, because those genes cut, splice, and rearrange themselves in unique combinations. You also may recall from Biology 150, alternative RNA splicing, where the introns get spliced out differently, and those create different messenger RNAs, which produce different proteins. So we do have the ability to produce a unique antibody for every unique antigen out there. And so what that means is you have one particular B cell capable of making that unique antibody. So you have a lot of different B cells. So here we're seeing we can produce up to 10 to the 15 different antibodies. That's a huge number, 10 to the 15, right? So that means you have at least 10 to the 15 different B cells in your body. So huge numbers. So we have the potential to create an antibody for any given antigen that exists. So that's pretty amazing when you think about it, right? The ability to shuffle genes and to create all those different mRNAs to produce all those unique antibodies. And this all happens during embryonic development, right? So during fetal development, the immune system is being created, right? It's being developed. So it's not ready yet, but that's what's happening. One of the things that's happening during that 9 months. So how do the antibodies get made? Well, we know that the B lymphocytes are the cells that produce the antibodies. We know B lymphocytes have IgD antibodies on their surface, and they each have a different IgD antibody specific for a specific antigen. In this example here, we see that our C B cell is the one that can actually respond to this antigen, not the others, but just this particular one. So when that happens, when the B cell encounters that specific antigen, the B cell gets excited, it gets stimulated. It will proliferate and differentiate into two cell types. So it multiplies and creates two different cell types. One cell type is the plasma cell. And the plasma cell is a B cell whose metabolism has shifted to the production of that antibody and almost nothing else. So the plasma cells are antibody factories. They are just turning out lots and lots of antibodies. And the reason why is those antibodies have to travel all throughout your entire body to encounter that antigen wherever it might be. So you have a lot of plasma cells all secreting antibodies. The other cell type produced is a memory B cell. Memory cells remember for later. So what that means is the next time you encounter that antigen, you have a lot more B cells that are ready to respond. And the kind of antibody they will make is not the big IgM, but IgG antibodies. And so they'll be able to make those antibodies so fast that you become immune, right, to that pathogen or to that particular antigen, assuming it's something you've encountered before. So when B cells get stimulated, they multiply, right, they proliferate. They form two cell types, memory cells and plasma cells, and the plasma cells are antibody secreting cells, right? Their entire metabolism is basically shifted over to antibody production. So they make lots and lots of antibodies. But then you have memory cells to remember for later, right, for the next time you encounter that antigen. But this process here that we're seeing is kind of a random process, right, where the correct B cell just happens to interact with that antigen. And that's not really the most efficient way to do things, right, to rely on a random encounter. And so this is where your helper T cell comes along with macrophages, with some type of antigen presenting cell. And so the helper T cells actually help to stimulate your B cells to make antibodies against that particular antigen. But that involves a type of phagocyte, mostly a macrophage, but also dendritic cells, another type of phagocytes. But even under certain circumstances, B cells can act like an antigen-presenting cell, but we're not going to get into that because that's one of those fine details we don't have time to go into. But the main idea is that you have an antigen-presenting cell, like a macrophage, or a dendritic cell, so some type of phagocyte. It wanders around, it finds the antigen, it swallows it, it digests it, and then it takes pieces of the antigen to a helper T cell, right, to say, look what I found. In the process, the macrophage will secrete a chemical mediator, interleukin-1, which stimulates that helper T cell. And the helper T cell will multiply, it'll proliferate, and it will differentiate and form memory T cells as well. And the memory T cells are there to remember for later. But the T cells, they will secrete interleukin-2, which not only stimulates the helper T cell even more, but it helps to stimulate the B cells to make the antibodies. So this is a more coordinated effort. And if we use military terms, you can think of the APC not as an armored personnel carrier, but the APC is performing reconnaissance. It's gathering intel. And it takes that intel, to headquarters, to the general, which is the helper T cell. And the helper T cell alerts, right, the military, right, the ground troops, the B cells, right, to produce antibodies, which you can think of as artillery, right, to attack and destroy those foreign antigens. But it's a much more effective, coordinated effort. And so here in this diagram, we do see the central role of a helper T cell. We have the macrophage. It finds the antigen, it engulfs it, it destroys it, and then it takes pieces of that antigen and presents them to the helper T cell to say, look what I found. In the process, the macrophage secretes interleukin-1, which stimulates the helper T cells. Right? So they'll multiply, and they'll make some memory T cells. But now you have a lot more helper T cells. And the helper T cells secrete interleukin-2, which feeds back on itself to stimulate the helper T cells even more. But also the helper T cells can now stimulate a B cell to make the correct antibody. Also cytotoxic T cells, which we'll talk about later, they also get stimulated. But this is a more coordinated approach, right, to stimulating the correct B cell, right, to make the correct antibody. So the macrophage has gathered the intelligence, it's taken it to the general, the helper T cell, so the general can alert the troops, right, the ground troops, and they can start firing their artillery, right, the antibodies. So what happens when there's no helper T cells? So what happens to your immune response when there's no helper T cells?
They can't stimulate the B cells even?
Well, so B cells, they can still be stimulated by direct interaction with antigen, but your immune system becomes crippled. You become immunocompromised. It's hard to mount an effective immune response without the helper T cells. And what disease eliminates helper T cells? HIV/AIDS, right? So helper T cells are the primary target for HIV. And that's why AIDS patients become immunocompromised and susceptible to all kinds of infections because they can no longer mount an effective immune response because their T cell count is so low. So T cells are extremely important in the overall functioning of the immune response. Now, since we're using military terms, sometimes there are spies, right? Sometimes there's an attempt at disinformation. So the idea here is, how does the helper T cell know that it can trust that macrophage? How do they know they're in the same army, on the same team, right, fighting the same enemy. The way they know that is that the macrophage presents the antigen along with the MHC molecule. And the MHC is the major histocompatibility complex. It's a self-recognizing molecule, so that way they know they're on the same team, right? The helper T cell knows that it can trust that macrophage because the macrophage has presented, the code word or the secret handshake, so they know they're on the same team. So the antigen is presented along with the MHC to a helper T cell. And the helper T cell gets all excited and stimulated, right? Interleukins get secreted, and that helps to stimulate B cells to produce the antibodies. So it's a more efficient way, a coordinated way for the immune system to make antibodies because the macrophages, they're wandering through your body, they find the antigen and they take it to the immune system, they take it to the helper T cell, right, to alert the immune system to start making antibodies against that antigen. So any questions on this diagram? So a few words about the antigen presenting cells, the APCs. Mostly we talk about macrophages and dendritic cells because they do the same thing. As I mentioned, B cells can sometimes act as an antigen presenting cell, but we're not really going to go there. So macrophages, they can get stimulated, again, by chemical mediators like interleukins. So here we have a resting macrophage. So it's basically just sort of chilling, right? Hanging out, but when it's been stimulated, notice the membrane is all extended. And so these macrophages are on the hunt. They know there's something they need to seek out and destroy. And so when you activate the macrophages, they're now on high alert and they go out looking, right, for the enemy to find it. A really interesting experiment that we can no longer do because it involves blood. But if you search the internet, you'll find some videos of this. But if you take some blood and you centrifuge it, you know that you separate out the cells. And if you've ever seen centrifuge blood, you know that the bottom thicker layer are the red blood cells. And right on top of that is a thin layer that's white. And those are actually the white blood cells. When you centrifuge them, the white cells form a white layer on top of the red cells. And you can pipette some of those white blood cells onto a slide with some live bacteria and observe them attacking and engulfing the bacteria. So it's a really fun experiment to do. No longer allowed to do it because it involves blood, so we can't do it. But there's videos available online showing that process. So, but it's really interesting to see. So how long does it take to produce antibodies? Well, it takes a little while. It's not instantaneous. So on average, the first time you encounter a brand new antigen, it takes your immune system about 7 to 10 days to start making antibodies. And remember, those are the big IgM antibodies. Later, your immune system will switch over to IgGs. But 7 to 10 days, so that's why when you get a cold or the flu, it takes about 2 weeks to recover because you may be infected for 1 to 3 days before you show symptoms. But your immune system is working, it's trying to make antibodies, but it takes a week to a week and a half to make those antibodies. And that's an average. In some people, it takes longer, but on average, 7 to 10 days. But after you've made those antibodies, you have memory cells, memory T cells, memory B cells. So they remember for later. So the next time you encounter that same antigen or that same pathogen, whether it's 2 weeks later, a month later, a year later, you can make antibodies in as little as a day. So you make antibodies so fast that you're immune, right? That pathogen can no longer cause an infection because you're able to make the antibodies a whole lot faster. So all subsequent exposures, you can make the antibodies in 1 to 3 days, so fast that it makes you immune. And normally for your entire life, you'll be able to do this. But when it comes to pathogens, we know they mutate and change, right, over time. So in fact, you may never encounter that particular pathogen ever again because it's mutated and changed enough. All the antigens are just different enough. Your old antibodies are no longer effective. So you have to get sick all over again, make more antibodies, and become immune. Over and over again, year after year after year for the rest of your life, right? Because viruses mutate and change. Bacteria can mutate and change as well. So that's why we keep getting sick, right? Because the pathogen has changed. Your immune system is still good, it's still doing its job, but it takes 7 to 10 days. If the pathogens mutate and change, you have to make antibodies all over again. Now, it's possible due to random chance that the pathogen may still be similar enough so that the antibodies you made can help. It can limit the infection, right? Have you ever had a very minor cold before? Maybe just a few days of sniffles and that's it, it's all done? It could be that you were infected with a virus you've had before and the antibodies, it'll still work well enough to limit the infection, right? So that could have been the possibility. If you've been watching or looking at the statistics for COVID-19 all over the world, what you may have noticed is that the infection rate in Africa is actually very low, and the death rate is very low in many African countries. And the hypothesis is that because coronaviruses are also causes of ordinary colds and respiratory infections, there may have been more of those kinds of respiratory infections in many African countries, and that has provided some protection against COVID-19. So antibodies those people already made against the harmless types of coronaviruses are helping to protect them against COVID-19. So once you make the antibodies, you have those memory cells, so you can make more. If the pathogen does not mutate that much, those antibodies will still help, right? They'll still be effective. And it could be, as in this case, certain coronaviruses where they just got colds, they have some immunity to COVID-19. So questions or comments? So we're gonna stop here today and we'll finish this lecture next time and do the hypersensitivity lecture. So we'll probably go longer next class to make sure we get finished with all this material. So, yeah, question, Rachel?
Yeah, just about Thanksgiving break. Is it just Thursday?
It's Wednesday and Thursday.
It's Wednesday and Thursday? So we don't have
Yes.
class next Wednesday?
Right, correct.
Okay.
Only on Monday. And then you get a break, unless you have Tuesday classes. Just Monday and Tuesday.
Okay, got it. Thanks.
All right, so I'll see you guys in lab around 10 after 1. All right?
Thank you.
See you soon.